Welcome to UPSC Vinoth. Today we are going to discuss a complete discussion about genetic science, all current affairs related topics with respect to genetic science. Genetic science is one of the most important topic of UPSC, most important topic for UPSC prelims. So in past 25 years, every year you will get at least minimum one question with respect to genetic science. Okay. So let us have a discussion regarding with genetic science. So for your information, I had already discussed about glow-cas9 glow-cas9 topic in previous video. So before watching this video, just go and look into this glow-cas9 video. Okay, it is a continuation of that genetic science, that is glow-cas9. Okay, so now after glow-cas9, the most important current affairs with respect to genetic sciences, DAISY gene editing technology. See, DAISY gene editing technology. So it is DAISY means what? Indian gene editing technology for cheaper genetically engineered crops breeding. For cheaper genetically engineered crop breeding. So this is in the news. So Indian scientists have developed a miniature alternative to CRISPR-Cas9. See, CRISPR-Cas9 is used for gene editing technology. So Indian scientists have developed a miniature alternative. So this makes this topic is more important because already there has been multiple questions with respect to CRISPR-Cas9. So now this question, this concept is going to be a more important for coming year prelims. So Indian scientists ICAR patented the developed miniature alternative to CRISPR-Cas9. So based genome editing to precisely cut and tweak plant DNA using TNBP, transponder associate protein. So Indian scientists have discovered a new gene editing technology. So that also seems to be cheaper. So that is called as TNBP, transpond associate protein. Transpond associate protein. Okay. So what is the new technology all about? So TNBP based genome editing. So see the topic, TNBP based genome editing. That is transposon associate protein. So TNBP, transposon associate proteins. So a new gene editing system using miniature TNBP protein as a molecular scissor. So they are using TNBP protein as a molecular scissor. Earlier, CRISPR CRISPR was used as a molecular scissor. Now they are going to use TNBP protein, which is like CRISPR. This is TNBP protein is also like a CRISPR. So this also used as a molecular scissor. So to cut the DNA. So this TNBP protein, is used to cut the DNA at predetermined targets. It is used to cut the DNA. It is a gene editing technology used to cut the DNA at the what predetermined targets. Okay, what is CRISPR-Cas9 technology? So this is already we had discussed earlier. It is like a revision. It is a gene editing system where it is guide RNA directs Cas protein to cut the specific DNA, cut or modify the specific DNA. So using Cas protein, so using CRISPR-Cas technology, by using Cas protein, it is used to cut, modify the specific DNA. A molecular scissor navigator pair enables targeted changes in gene. So it will enable the targeted changes in the gene for research, agriculture, and therapeutic purpose. Okay, what is the new technology all about? Now TNPB technology deploys TNPB protein. Now we are using, see, instead of using CRISPR protein, now we are using TNBP protein as a molecular scissor. See, as a molecular scissor. So see, TNBP protein is a molecular scissor. So cleave DNA at a predetermined targets. To cut the DNA at a predetermined targets. Predetermined targets. To TNBP protein is used to cut the DNA targets at a cut the DNA at predetermined targets. It is very small. It is very small. Source from extreme surviving hypercompact 408 amino acids. See, it is very, very smaller than CRISPR. So it is very, very smaller than, when compared to CRISPR, this TNBP protein is very much smaller, functioning with high precision. So when compared to CRISPR Cas9, it is functions with high precision. It is very much important, very much important. Okay, so advantages of TNBP protein. What is the advantages? See, it's comparing. You should compare with the earlier process, earlier technology. TNBP is made up of, it is size of 400 to 500 amino acid, but Cas9 and Cas12a, this two proteins are 1000 to 1400 amino acids. So easier delivery. So TNBP has some easier delivery in plant cell. See, plant cell, they will all see how they will ask, like, TNBP protein TNBP protein can be used to deliver cells easier in animal cell. No, in plant cell only. Hypercompact edited. Hypercompact genome editors. So hypercompact genome editing is possible only. Hypercompact genome editing is possible only in TNBP. So it is not present in Cas9. Delivery method, direct injection. It is very easier to viral vector packing. Direct injection. You should culture in tissue. You should culture in tissue for Cas9. So we already know about the Cas9. We should culture in tissue and then we should deliver. It is like we can deliver directly. See, TNBP, that is TNBP is transposon associate protein. This is new molecular scissor. This is a new molecular scissor. It is updated easier when compared to Cas9. So it is easier to deliver into the plant cell. So hypercompact genome editor. So it can be delivered directly. So what is the advantages? Finally, we are coming to the question part where the UPSC will focus. Potential impact, cheaper and faster in crop. See, they are mainly looking at crop via TNBP. Agriculture, faster breeding. Precision gene tweaking for decided traits. So gene editing for decided traits. Cheaper. So reduced cost of genetic engineering crops. So it reduces the cost of genetically engineered crops. So this video is brought to you by UPSC Vinoth. Myself is Vinoth Kumar. So you can follow my channel UPSC Vinoth in my to get more current affairs updates and MCQs. So with respect to climate, it is stress tolerant. It's a climate resilient. See, these three are very much important. So climate, economic, agriculture. Climate, economy, agriculture. Okay. So see, we have now we will discuss the MCQs with respect to TNBP based genome editing technology. So already we had discussed all PYQs in CRISPR Glow Cas9 video. So now we will discuss only the module MCQs. TNBP based genome editing technology. First question. Consider the following statement about TNBP based genome editing technology. So first statement, TNBP protein acts as a molecular scissor similar to Cas9 and Cas12a. Yes, statement 1 is correct. Yes, you know that it is acts as a Cas9 and Cas9 and Cas12a. It enables targeted DNA modifications in plant. Yes, it enables targeted DNA modifications in plant. It requires a tissue culture mediated delivery. No. Which requires tissue culture mediated delivery? CRISPR-Cas9 requires tissue culture mediated delivery, but it is like direct injection. Delivery method is direct injection. See how I am recalling the points. With respect to MCQs. With the help of MCQ, you can crack this exam. Yes, this is one of the most quality MCQs which is provided by myself, that is Vinoth Kumar. So TNBP protein cuts DNA like Cas protein, allows precise editing but do not require tissue culture. Unlike CRISPR-Cas9, it direct injection. Okay. Which bacterium is a source of hypercompact TNBP genome editor? So this is one of the important thing. Deinococcus radiodurans. Deinococcus radiodurans. This bacteria is used to So which bacterium is the source of hypercompact TNBP genome editor? So the TNBP genome editor is taken from Deinococcus, Deinococcus radiodurans. Deinococcus radiodurans. TNBP is used in this technology is a source from Deinococcus radiodurans, a bacterium capable of surviving in extreme environments. See the application. This TNBP will also serve in extreme environment. One of the major advantages of TNBP protein over Cas9 is what is it is smaller size enables easier delivery into the plants. It is smaller size. So one of the major advantages of TNBP project is one of the smaller size enables easier delivery into the plants. So which of the following statement is about CRISPR-Cas9 genome editing is correct? Which of the following statement is about CRISPR-Cas9 genome editing is correct? So see, it is used as a guide. It is old. It is used as a guide RNA to direct Cas9 protein to specific DNA sequence. Yes, it is uses a guide RNA. It is uses a guide RNA to direct Cas protein to specific DNA sequence. Okay. It is functions like molecular scissors. Yes, it functions like molecular scissors. It cannot be applied in agriculture. No, it can be applied in agriculture. So 3, 1 and 2 only correct. Which of the following features makes TNBP suitable for viral vector delivery in the plant? See the term viral. See how the questions are made. Viral vector delivery. I'm constantly using the keywords which has been discussed in the current affairs content so that you can remember and recall the points. Viral vector delivery. TNBP is suitable for viral vector delivery. Why? Why? So it is smaller in size. Yes, when compared to Cas9, it is very smaller in size because 1000 to 1200 is reduced to 400 to 500 amino acids. Hyper compact structure from extreme mobiles. So it is hyper compact structure. Yes, ability to bypass tissue culture mediate delivery because we are using direct injection method. No tissue culture is used. Adaptability to bypass. So 1, 2, and 3 both all are correct. Okay. Which of the following is not true about TNBP genome editing? Which of the following is not true about TNBP genome editing? See, it can precisely edit the DNA like CRISPR-Cas9. Yes, it can precisely. They are asking not true. It requires larger amino acid size. See, it requires smaller amino acids than Cas9. So B, this is correct. Means this is wrong. This is not true about TNBP editing. It can deliver via viral vector. Yes, it can deliver via viral vector. See how I am using the keywords repeatedly so that you could not forget the thing when it is asked in the exam. So it is hyper compact system delivery from extreme mobile bacterium. Yes, it is a hyper compact system derived from high extreme mobile bacterium. Yes, all three are correct. This is wrong. It is smaller amino acid. So TNBP is smaller than Cas9 and Cas12, which is one of the key advantages. CRISPR-Cas9 statements. Uses guide RNA to direct. Yes, it is uses guide RNA. So again, function as a molecular scissor, cannot apply in agriculture. So it is a repeated question. Okay. So this is very much important topic. Yes. So see, if you are means this is one of the most comprehensive discussion which is made via with the help of MCQs. Okay, let us move to the next topic. Organ on chip technology. See, first of all, you must know what is meant by organ on chip technology. Just to syllabify the word organ on chip. So here we are seeing that organ on chip. So why it is in the news? So it can transform the drug delivery. See, this organ on chip can, one second, this organ on chip is technology is mimicking the human body for better drug development. See, mimicking the human body for better, for what, for better, for organ on chip is a mimicking the human body for better drug delivery. So it has transformed the drug development. It has transformed the drug delivery system, which is already existing in this in the medical history. So it is one of the very much important topic. So I choose this topic because it is transforming the past. So now organ on chip. See, organ on chip. See, OOC technology. What is meant by OOC technology? So it is a micro scale system. It is a micro scale system. First of all, it is a micro scale system mimicking the human body environment. See, organ on chip is refers to micro scale system. It is a system, so it is mimicking the human body development. So see, it is one of the human relevant 3D culture model. So it is new approach. So new approach method, it is also known as new approach method. OOC method is also known as new approach method. What is this? 3D culture system allows recreation of human organs. See, they will create a human organ in one dish, and another, and two, and two diseases on one dish. See, it is like you to test the disease. It is recreating the human organ one in one dish and diseases in one dish. Now they will put the diseases on the human organ, and they will test the how the disease is reacting with the human organ, so that they can produce cure to the disease. So now see, it breaks one of the greatest hurdle, so that you can see the medicine can't be tested in humans. So now, with the help of this organ on chip technology, you can make a human-like 3D culture system like organ, and also you can test with the diseases and whatever drugs can be tested with the particular disease. Okay, so human relevant, more accurate than animal model. So generally we will test the drugs on animals, but now this system bypasses this, and it makes the human like 3D system, so that it can be easily tested, and the result can be precisely that is equal to what how it can be means it is a result that it is similar to how it tested in humans. The OOC mechanism, how it works. See how it works. Cell placement. Specific cell are placed on the chip. So it is placed on the chip. Polymer matrix. Polymer resemblance of real tissue. 3D growth. The cell allows to growth in 3D structure. Human-like organ cluster. Cells are placed on the chip and allowed to grow on 3D structure with the help of polymer that resembles the real tissue in human body. Okay. What are all the benefits of organ on chip? See, what are all the benefits? So see, it's very easy. So human relevant ethic. So whenever the ethics comes, you can't test the drugs on humans. So now we can test the drugs on what? Organ on chip. Ethical, cost effective. Provide human relevant platform, accurate drugs. So next, personalized medicine for enabling patient specific treatment. So by this we can, for liver, for lungs, for heart. So for lungs, for heart. So whatever it may be, you can be precision medicine. Precise medicine can be tested on the particular organ by using this organ on chip method. See, by using this organ on chip method. So see, you people will get little bit confused by discussing with the MCQs. You will get a clarity. Which of the following is a primary advantage of OOC? See, OOC over conventional method. See organ on chip over conventional method. Enabling high cost. See, you know that it is low cost. So primary advantages. What is the primary advantages? Which of the following is primary advantages? See, what is a primary advantage? Provide human relevant and ethical cost effective platform. See, it is provide human human relevant ethical cost effective platform. It is a primary advantage. Okay, providing human relevant ethical cost effective platform. OOC reduce the reliances on animal module, allows accurate drug testing. So instead of testing on animal, we can test this in this model. Organ on chip can be implemented in what are all the following? Drug testing, disease modeling. So disease modeling medicine, space travel, logistics, everything. See, it is 1 2 3 1 2 3. See, all these things, all three things. So OOC supports drug testing, disease modeling, and personalized medicine for unrelated and unrelated to, sorry, this is 4. It is not related to space travel logistics. Sorry for disturbance. So I had made a mistake. So here this is 4. So here the fourth option is not there. So it can be drug testing, disease modeling, personalized medicine. So it is personalized medicine. Personalized medicine. So 1, 2, 3. It's not used in space travel logistics. So which of the following is true with respect to OOC? See, which of the following is true? It is a 2D. So it is 3D. It uses microscale system to mimic human. It eliminates the need of any laboratory experiment. No. It is exclusively used for plant tissue research. See, it is used for plant tissue research. Okay, fine. Organ on chip is a category under what? It is a new approach method. New approach method in human relevant 3D culture. Sorry. Here it comes under microscale system mimicking human tissues. Sorry. I had made a mistake at the time. So it is used as microscale system of mimicking human tissues. Okay, fine. So this is very much important topic. So I think that I gave a clarity with respect to organ on chip. So it is in the news. It can transform the drug delivery. Microscale system used for mimicking the human body environment. See the keyword which has been replicated in the questions itself. Microscale system used in the mimicking the environment. New approach method in 3D culture. 3D system recreates human organ. See, it is precisely I mentioned every important point. Okay, so it is human relevant, ethical, cost effective. Accurate drug testing, disease modeling, personalized medicine can be created. So these are all very much important points. So I think I am this discussion is helpful to you. Okay, fine. Kamala and Pusa, this seems to be, yes, this also having a high potential to come in this exam. So why? Because it is rice. Rice is our staple food. In UPSC, just go and check previous year question paper. Rice has been one of the important theme in past 25 years. Every year, you will get minimum one question with respect to rice. Every year, you will get minimum one question with respect to rice. Yes, I'm damn sure about that. If they are looking for rice, yes, there will be a question. Kamala and Pusa DST Rice 1. World first genome edited rice. Kamala and Pusa. So it is world first genome edited rice. It is developed by ICAR. Okay, fine. There are two types of genetically modified rice. Kamala and Pusa. Okay, fine. Why in the news? Fine. So ICAR developed Kamala and Pusa. Who developed ICAR? ICAR developed Kamala and Pusa using the CRISPR-Cas9 genome editing to enhance yield, drought, and salinity tolerance. To enhance yield, drought, salinity tolerance, and nitrogen use efficiency without any foreign DNA. See, this is the extreme keyword, but this can be asked in the exam. This Kamala and Pusa DST rice has been it is a genetically modified rice using CRISPR-Cas9 technology, and it is done using without any foreign DNA. It is not. It is like gene editing. See, it is. See, they are using the word genome editing. See, they are editing the gene. They are not inserting anything new. They are just editing the gene. Okay, fine. So it is editing. Okay, fine. So here, without using any foreign DNA, this is one of the important key point. So this is one of the important key point. ICR developed world first genome edited rice varieties, DRR. So DRR. So it is one of the important thing. So important thing. So DRR Dhan 100 Kamala. So Pusa DST rice 1. So it is developed by ICAR IIRR Hyderabad. It is developed by ICAR IARI New Delhi. So Kamala is developed by Hyderabad. Pusa is developed by New Delhi. Kamala is developed by Hyderabad. They will interchange it. What? Kamala is developed by New Delhi. Pusa is developed by Hyderabad. So yes, UPSC has a what there's a nature to swap the options, swap the content. Parental variety of Samba Masuri here. It is parental variety of MT. See, these are all not very important. So just look at this thing. These two are very much important. Targeted gene. So gene targeted cytokinin. So here gene targeted drought and salt tolerance. So technology. So these are all SDN1 technologies. This also SDN1 technology genome editing. See, these are all not very much important. See, what are all the advantages? So why this genome editing has been done? See, genome for this early maturity. So they are looking for early maturity, drought tolerance, high nitrogen use efficiency. See, high nitrogen use efficiency. Superior yield 5.37 tons per hectare, 19% has been increased. So yield has been increased. See, early maturity than earlier 20 days earlier. So it normally takes 150 days. Now it takes 130 days. So you get 20 days earlier. So superior yield, the yield is getting high quality. 19% has been increased in the yield. That means 5.37% ton per hectare has been increased. So with compared to parental varieties. So and also it read as the grain cooking quality, and also it read as the grain cooking quality also. So see these areas, this DAR 100 can be recommended to zone 8, 5 and 3, that is Andhra, Telangana, Karnataka, Tamil Nadu, Puducherry, Kerala, Chhattisgarh, Odisha, UP, West Bengal, Bihar, Jharkhand. So these are the recommended zones. See, these are all not very much important, but just have insights on this. Next, coming to Pusa. Pusa is developed by ICAR, IARI, New Delhi. Yes, you know that parental variety gene targeted is drought and salt tolerance. Here high yield, drought tolerant, high nitrogen use efficiency. So SDN1 genome, both SDN1 genome editing technology. Key traits is drought and salinity tolerance. So key traits, here's a key traits also, drought and tolerance, not drought tolerance. Here, see, salinity tolerance has been not discussed in Kamala, but in Pusa salinity tolerance has been implanted. Okay, so coastal salinity, particularly coastal salinity. Okay, recommended like major rice growing states across India. It is they gave commonly major rice growing states across India. Okay, fine. Okay, so inland salinity is 3,508 kilogram per hectare. Alkaline salinity, see, these and all not very much important. About site director nuclear test. SD1 introduced a small insertion delegation without foreign DNA. Yes, we know that. SDN2 using template of DNA similar to host introduced to specific changes. A precise DNA cutting technique is used in nucleus. Okay, fine. So now we are coming to the MCQ section. Consider the following statement regarding recently developed genome editing rice variety. DAR 100 Kamala is developed by ICAR IARI. So DAR 100 has been developed by ICAR IARI. No, you know that IRRI. So Indian Rice Research Institute. So this is statement 1 is wrong. The DST1 rice targeted CKX2 gene, X2 gene to confer drought and see it is targeted which gene. So I didn't discuss about this gene. So this is also wrong. Both varieties were developed using SDC, site directed nucleus 1 technology, SDN technology, site directed nucleus 1 technology. Yes, both the varieties are, when compared to Pusa and Dar, both DRR done, both varieties are developed using site directed nucleus. Yes. So containing no foreign DNA. Yes, containing no foreign DNA has been introduced. Okay. So statement 1 is incorrect, statement 2 is incorrect, statement 2 is correct. Okay. Second question. With reference to genome editing rice variety, DRR, DAN 100, Kamala, which of the following statement is correct? See, parental variety is MTU1020. This is for what? See, UPSC generally won't ask this question, but it is MCQs. So you this is used for PUSA. MTU 1010 is used for PUSA. MTU 1010 is used for PUSA. So here is Samba Masuri. Here the parental variety is Samba Masuri. See, it's comparing with when you are comparing with Kamala, it is Samba Masuri. So it is Pusa. For parental variety M10, MTU 1010 is Pusa. Gene targeted. So with reference to genome editing rice variety, DAR is gene targeted is DST. No, just now we saw that gene targeted is CKX2. It is not DST. So DST is for what? PUSA. Again, DST is for PUSA. Key traits is salinity tolerance. So see, key traits is salinity tolerance. See, no drought tolerance. So salinity tolerance is for what? PUSA. Again, salinity tolerance is for yield is 19% has been increased. Generally, UPSC won't focus on data, but to recall the data, I had made like this question. Okay, fine. Consider the following statement regarding ICAR, DAR, and PUSA, and both varieties. This is the question. This question is much valid question for UPSC. Both were developed using CRISPR-Cas9 to target gene for drought salinity tolerance. DAR 100 retains Samba Masuri grain quality and is suited for zone 7 states like Tamil Nadu. DAR 100. PUSA DST Rice 1 outperforms MTU 1010 only in drought, not in salinity condition. See again the comparison is both. So see, PUSA DST outperforms in MTU only in drought condition because PUSA MTU is for salinity condition. It is for salinity condition. So 1 and 3 is correct. 1 and 3 is correct. So yes, 1 and 3 is correct. Okay. So this is very much important. This is very important current affairs. So let us move to next topic. National Biobank. National Biobank. So let us see the National Biobank in next video. Thank you. Keep following. So let surely you will get one question from this and upcoming video. Watching 2 videos will fetch you 2 marks. I'm damn sure about it. Thank you.